Hey everyone, let's start the NCERT audiobook 2023 edition. Here the first chapter is sexual reproduction in flowering plants. Now some introduction. Biology in essence is the story of life on earth. While individual organisms die without fail, species continue to live through millions of years unless threatened by natural or anthropogenic extinction. Reproduction becomes a vital process without which a species cannot survive for long. Each individual leaves its progeny by asexual or sexual means. Sexual mode of reproduction enables creation of new variants so that survival advantage is enhanced. This unit explains the details of reproductive processes in flowering plants and humans as easy to relate representative examples. A related perspective on human reproductive health and how reproductive ill health can be avoided is also presented to complete our understanding of biology of reproduction. Pancham Maheshwari, born in 1904 in Jaipur, Rajasthan, Pancham Maheshwari rose to become one of the most distinguished botanists not only of India but of the entire world. He moved to Allahabad for higher education where he obtained his DSc during his college days. He was inspired by Dr. W. Jason, an American missionary teacher, to develop interest in botany and especially morphology. His teacher once expressed that if his student progresses ahead of him, it will give him a great satisfaction. These words encouraged Panchanan to inquire what he could do for his teacher in return. He worked on embryological aspects and popularized the use of embryological characters in taxonomy. He established the Department of Botany, University of Delhi as an important center of research in embryology and tissue culture. He also emphasized the need for initiation of work on artificial culture of immature embryos. These days, tissue culture has become a landmark in science. His work on test tube fertilization and intra-ovarian pollution won worldwide acclaim. He was honored with Fellowship of Royal Society of London (FRS), Indian National Science Academy, and several other institutions of excellence. He encouraged general education and made a significant contribution to school education by his leadership in bringing out the very first textbook of biology for higher secondary schools published by NCERT in 1964. Now, Chapter 1, Sexual Reproduction in Flowering Plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we soon over, the rich colors that attract us, are all these as an aid to sexual reproduction. Flowers do not exist only for us to be used for own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structures of the inflorescence, flowers, and floral parts shows an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the processes of sexual reproduction in flowering plants. Angiosperm. 1.1 Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since the time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbols for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of 5 more flowers that are used in social and cultural celebrations in your family. Have you heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In earlier classes, you have read the various parts of a flower. Figure 1.1 will help you recall the parts of a typical flower. Can you name the 2 parts in a flower in which the 2 most important units of sexual reproduction develop? Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, The decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a whorl of stamens representing the male reproductive organ and the gynoecium represents the female reproductive organ. 1.2.1 Stamen, Microsporangium and Pollen Grain. Figure 1.2a shows the two parts of a typical stamen, the longer and slender stalk called the filament and the terminal generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen each from 10 flowers, each from different species, and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and making neat diagrams would elucidate the range in shape and attachment of anthers in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are dithecous. Often a longitudinal groove runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is a four-sided tetragonal structure consisting of four microsporangia located at the corners, two in each lobe. The microsporangia develop further and become pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. A structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers: the epidermis, the endothecium, middle layers, and the tapetum. The outer three wall layers perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possess dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells would become binucleate? When the anther is young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspores or pollen grains are formed that are released with the dehiscence of anther. Pollen grain. The pollen grains represent the male gametophytes. If you touch the opened anthers of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water taken on a glass slide and observe under a microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent two-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzyme that degrades sporopollenin is so far known. Pollen grain exine has prominent apertures called germ pores where sporopollenin is absent. Pollen grains are well preserved as fossils because of the presence of sporopollenin. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative and generative cell. The vegetative cell is bigger, has abundant food reserve, and a large irregularly shaped nucleus. The generative cell is small and floats in the cytoplasm of the vegetative cell. It is a spindle shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this two-celled stage. In the remaining species, the generative cell divides mitotically to give rise to the two male gametes before pollen grains are shed, the three-celled stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders, asthma and bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grains lose viability within 30 minutes of their release, and in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. You may have heard of storing semen sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen minus 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. The pistil, megasporangium ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistils may be fused together, syncarpous, or may be free, apocarpous. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated cylinder part beneath the stigma. The basal burst part of the pistil is the ovary. Inside the ovary is the ovarian cavity locule. The placenta is located inside the ovarian cavity. Recall the definition and types of placentation that you studied in class 11. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be 1, wheat, paddy, mango, to many, papaya, watermelon, orchids. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule, figure 1.7d. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle the nucellus except at the tip, where a small opening called the micropyle is organized. Opposite the micropylar end is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant reserve food materials. Located in the nucellus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of four megaspores. Female gametophyte. In a majority of flowering plants, one of the megaspores is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy of the cells of the nucellus, MMC, the functional megaspore and female gametophyte? Let us study about the formation of embryo sac in detail. The nucleus of the functional megaspore divides mitotically to form two nuclei which move to the opposite poles, forming the two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of the four nucleate and later the eight nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. After the eight nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac. Six of the eight nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have special cellular thickenings at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. Three cells are at the chalazial end and are called the antipodals. The large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryo sac at maturity, the 8-nucleate, is 7-celled. 1.2.3 Pollination. In the preceding sections, you have learned that the male and female gametes in flowering plants are produced in the pollen grain and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen, pollination can be divided into three types. 1. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther to the stigma of the same flower. In a normal flower which opens and exposes the anthers and the stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity, and also the anthers and the stigma should lie close to each other so that self-pollination can occur. Some plants such as viola, common pansy, oxalis and commelina produce two types of flowers: chasmogamous flowers, which are similar to flowers of other species with exposed anthers and a stigma, and clistogamous flowers which do not open at all. In such flowers, the anthers and stigma lie close to each other. When anthers dehisce in the flower buds, pollen grains come in contact with the stigma to effect pollination. Thus, clistogamous flowers are invariably autogamous as there is no chance of cross pollen landing on the stigma. Clistogamous flowers produce a assured seed set even in the absence of pollinators. Do you think that clistogamy is advantageous or disadvantageous to the plant? Why? 2. Geitonogamy, the transfer of pollen grains from the anther to the stigma of another flower of the same plant. Although geitonogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grains come from the same plant. 3. Xenogamy. Transfer of pollen grains from anther to the stigma of a different plant. This is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. That's all for today. Hope you like it. Please like and subscribe for more such videos. Thank you.